Hello, good morning. Next Dr. Ben update is a paper from Dennis Burton's group. And I should mention my shirt first. You can see there's little tiny pink and green fish on there. They kind of look like little watermelons that we were wearing yesterday. I think they're maybe meant to be rainbow trout from the coloring on there, or maybe some kind of reef fish that I don't know. But if they're rainbow trout, the fin on top is wrong. I think trout have a fin that looks kind of like that, and these fish have kind of a sail-like fin, more like you'd see on a cichlid fish than a trout or a salmon or something. So maybe I just don't know what fish this is, or maybe it's just a cute shirt. I don't know. Who can tell? But wardrobe by Nicola, as always. Okay, so this paper from Dennis Burton's lab. And so Dennis Burton's thing, as long as I've been paying attention, has been antibodies. Antibodies to fight dangerous diseases, antibodies to save the world, antibodies you could never even imagine, existing. And yet there they are. And finding out the structures of antibodies and exactly how they work and stuff like that. So Mr. Antibody, and what do you know? This paper is about antibodies. Yeah, go figure. But it's a good paper. They're all really good papers, and he publishes in fantastic journals, and he publishes with these giant papers with like 30 people on them. This one's no exception. And it's a preprint in bioRxiv. And I would imagine this is going to either Nature or Science. He probably gets to pick at this stage. Maybe PLOS pathogens if he has to slum it. What he's done is taken antibodies from an infected hamster and grown them up in a lab and then made large quantities of these. They then uses to protect another hamster. And this is an idea that could potentially be transferred to humans. Doesn't have to be hamsters. There's nothing about this that would only work with hamsters and would not work with humans. So let's have a look. All right. So in the paper, what he does is he takes the first hamster, gives it the spike protein, because it's always the spike protein with these vaccine studies, yeah. And the reason for that is that it's the only thing that actually sticks out. So you've got your virus and the spike protein sticks out like that on the top. And so it is visible to antibodies, and most of the other virus proteins are not. And so he takes the spike protein, puts it in, the hamster makes a nice response against the spike protein, and now the hamster has antibody-producing cells. And the thing with these antibody-producing cells, they'll make something like a copy of the antibody that's actually stuck to the surface. And they'll also start later on releasing some of these antibodies once they get really good, once they have a definite match. He's using the antibody stuck to the surface as a way to sort out the good cells from the bad cells. Because you have lots of antibody-producing cells in your body at any one time, but only a few of them are actually making useful antibodies. And so he's just using the spike protein. He's got all the cells, and he's sorting them out by which ones can actually stick to the actual part of the spike protein of the virus that is the one that causes entry. And it's one of those three, remember there's three little things on the top. One of them sticks up at a time and grabs onto the receptor. That's the part. The receptor binding domain, it's called. And so that's clever. And then what his team do is they copy out the genes that that cell had made, which are like the instructions for making that antibody that can actually bind to that part of the protein. And then just does this blindly, just copies all the ones that came out, and then grows all of these up inside of new cells, and makes lots and lots of them, gives it 3 days to just produce a bunch of antibodies. And then now that he's got a bunch, he takes these antibodies and he puts them into other hamsters that have been given the virus, and shows that he can actually knock out the virus and save the hamster. Now, will this knock out the virus and save a human? We do not know. Some of these antibody tests seem to work better in little rodent models than they do in human models. Yeah. From what we've seen with previous viruses like the antibodies against Ebola and several other kinds of virus, where the antibody tests looked real good until we got up to people, and then not the antibody tests but antibody treatments, and then in people we're a little bit hit or miss. We're big. We're big. We got a lot of real estate. It's hard to get enough antibodies everywhere. You just got to make a ton of them. Anyway, it's an interesting paper. It's an interesting strategy. It's pretty quick, and it's using the actual immune defenses of people. It's a way to use the actual immune defenses of people who have got COVID and survived it to actually protect other people. Maybe, yeah. So yeah, stay tuned. We'll figure out if this works. I'm sure they're already working on the next step, trying to get this to something like clinical trials. Thanks very much. This is